and I'm co-convener of the BRLSI Science Group. A very good evening to you all, friends and members of the BRLSI, and all of our guests this evening. Before we begin this evening's business, I'll just remind you that during this live Zoom talk, we're going to ask you to keep your microphone on your computer or tablet muted, and also to make sure to switch off the camera. The slider controls to do this should appear at the bottom of your Zoom screen. And thanks in advance for doing this. Muting and veiling yourself will help preserve the bandwidth of the Zoom transmission so that the quality of the video and the sound will be as good as it can be for everyone to enjoy. So let's take a moment just to make sure that all those microphones and cameras are off. And by the way, we're going to be recording the proceedings so that if you don't want to appear even in a tiny way in the recording, just make sure that your video camera is turned off. As we go along this evening, questions that you might wish to ask may occur to you. And indeed, that's one of the things that we all like best about these evenings at the BRLSI. And the way that we're going to handle those questions is this. Please note that there's a button on your screen marked chat. And if you press this, it'll open up a dialog box for you to type your question. And the default is to address your question to everyone. That's what happens if you just type. And that means that others who are interested in looking at the chat screen will be able to see that your particular question has been asked. And this is good because it'll help us to avoid asking the same questions more than once. So type your question in as brief a format as you can. And we won't deal with it straight away, but at the end of the talk, there's going to be a question and answer session. And we'll take your questions one by one, and we'll do that in what seems to us like the most sensible order, because, of course, we can follow threads of thought if we do it in that way. And if a question appears to have been asked twice, we may just choose one of them, for example. And when we're doing the question and answer session, I'll ask you to unmute and to switch on your camera, unveil yourself, and ask your question in person at that time. That's always the most fun way to do it. Okay, so now we know what we're going to do. Well, tonight it's my privilege to introduce Dr. Tiffany Taylor. After a BSc in Evolutionary Biology at Edinburgh, Dr. Taylor did a PhD at the University of Oxford. And from there, she went on to the University of Reading, first as a postdoctoral fellow and then as a teaching fellow. And since 2016, she's held a prestigious Royal Society Dorothy Hodgkin Research Fellowship at the Milner Centre for Evolution here at the University of Bath. Her research concerns the evolution of novelty, literally how evolution just doesn't tweak things so that they work better if the circumstances change, but how sometimes completely new features arise. And as you'll see in a minute, this involves doing some experiments at the laboratory bench. So here, evolution isn't just some vague abstraction, but something that you can actually watch happening in real time. So I'll let Dr. Taylor introduce her own title, but before I do, I'll just mention that she's a phenomenally energetic person who's a keen communicator about science and why it's important. She's a regular reviewer of scientific books for the Times Higher Education, and she's also the author of 2 excellent children's books about genetics and why it matters. So, over to you, Tiffany. All yours.
Thank you so much for the lovely introduction. Hopefully you can see and hear me okay.
Indeed, we can.
Excellent. Okay, then I'll begin. So it's my real pleasure to be here. It's a shame we can't all be here in person, but I'm very grateful for everyone who's given up their evening to listen to me talk. So the title of my talk is Replaying the Evolutionary Tape. So this sort of explores the idea of what happens if you rewind the tape, the evolutionary tape, and start again in some random historic point and let the tape rerun. Do you end up in the same point again, or do you end up in a completely different place? And I'm going to explore ways that we can actually experimentally test this. And this is hence natural selection in a test tube. And this is using a method called experimental evolution. And I'm just going to give a little bit of an overview of what that means and the history of it as well. Oh, the mouse isn't working. Okay, so there we go. So what would happen if the hands of time were turned back to some arbitrary point and evolutionary history was allowed to rerun? So American paleontologist Stephen Jay Gould proposed this famous thought experiment in the late 1980s. And it's a question that still captures the imagination of many today. So Gould reckoned that if time was rewound, then evolution would drive life down a completely different path and humans would never re-evolve. In fact, he felt that humanity's evolution was so rare that you could replay the tape of life millions of times over and we wouldn't see anything like Homo sapiens arising again. And his reason that he believed this was because changing one small detail in history is likely to lead to larger consequences later on, because these consequences magnify over time. And chance certainly does play a huge role in evolution. So these can include big things like mass extinction events caused by cataclysmic asteroids or volcanic eruptions. But also chance operates at the molecular scale. So genetic mutation, which is the raw material in which evolution works with, is reliant on chance events. So just this week, the Mars 2020 mission landed the rover Perseverance on Mars. And the mission aim is to seek evidence for ancient life, which is really exciting. But if evidence is found, how likely is it that such life will seem familiar to us? So it goes sort of with reasoning, with logic, that any kind of organism that they found would need some kind of form of information storage and replication mechanism. So for us, that's DNA and RNA. But would it have the same? Would it have some sort of analog of that? And it's interesting to think about, because historically Mars actually shares many features with planet Earth, things such as the sort of radiation levels, atmospheric levels, liquid water. So we might expect some form of convergence. So although this opportunity to replay the tape of life on another planet is fascinating and hugely exciting, it's also expensive and risky in terms of its likely success. So there isn't enough money for everybody to go sending rovers off to Mars. So perhaps we need a less adventurous option to be able to sort of further explore these ideas. So the thought experiment from Stephen Jay Gould leaves us with a number of open questions. Things like, what are the factors that determine evolutionary outcomes? Are these factors ever predictable? And is there a way we might experimentally explore these questions? So I want to spend some time thinking about this third question more in detail now, and then I'm going to get to these two other questions a bit later in the talk. So to answer that third question, what we need to be able to do is study evolutionary processes. So what methods do we have to enable us to do that? Well, perhaps one of the first things that comes to mind is using the fossil record. So fossils can show us life forms that no longer exist, and they can offer insights into so-called missing links. So for example, this image of the well-known Archaeopteryx is thought to be important in the transition of dinosaurs to modern birds. So from fossils, we can gain an understanding of how long life has existed on Earth, when certain lineages appeared, how and where they lived. And it can also provide knowledge about ancient environments, so how climates have changed throughout evolutionary history by looking at the plant and animal type distributions. So fossils can tell us loads about extinct life, and also when certain major events occurred that might have altered the trajectory of certain evolutionary outcomes. However, fossilization is limited by many factors. So the conditions must be perfect to allow fossilization, and as such there isn't an even representation of all animal life across the fossil record. Hard-bodied animals are much more likely to successfully fossilize compared to soft-bodied. And the processes and drivers of adaptation have to be inferred from patterns. So getting a really detailed picture is difficult. So what other ways are there that we might be able to study evolution? Well, comparative anatomy looks for similarities and differences between species that might tell us whether they have some sort of common ancestry. So for example, here, if we're looking at humans, dogs, birds and whales, what we can do is we can look at their forelimbs. And if you look at the forelimbs, you can see that they share similar structures in terms of the bones that they carry in their forelimbs. And although they look very different, and this is because they are adapted to very different roles, the fact that they share these structures means that they are likely to have a common origin. However, we can run into problems with convergent evolution, and this is where organisms under the same selective pressure can evolve to look the same. And also, as structures become more complex, identifying true similarities and differences just becomes more difficult. So although this is another really useful tool, we need to still infer the relationships between organisms based on correlational evidence. So comparative phylogenetics is another method. So this is where we look at differences either in traits, so here the tails of different organisms, or maybe also looking at differences within the DNA. And what we try and do is look again at the differences and the similarities to try and identify possible relationships between species. And actually with looking at DNA evidence, what we can also do is we can add a time associated with some of these divergences. So based on the number of mutations that we see, we can sort of date the likelihood of when these divergences might have occurred within a certain time frame. So it's quite a powerful method. But again, all the methods I've discussed so far, they are very essential to contributing to our knowledge and understanding of evolution, but in each case, they require a number of assumptions that rely on correlational data, where we need to make predictions about historical events that we couldn't have been there to experimentally test. So is there another way where we might be able to run hypothesis-driven experiments that allow us to test for causation rather than relying on correlational data? So to do that, we need to observe evolution in action. So when we think of evolution, we typically think of a process that takes millions of years, but in fact, the rate of evolution is a factor of generation time. So organisms with fast generation times evolve faster. So let's take a look at some generation times. So if you wanted to observe the evolution of trees, we're looking at about 100 years per generation. And you need to observe many generations in order to see evolutionary change in action. So that's not really possible. So for humans, you're looking at around 20 years per generation, which is again, not really possible to see anything within a time frame of a typical scientific career. So mice, we're getting a bit more reasonable. So we're looking here at about 2.5 months. But still, even a trait under very strong selection, were you to give it 100 generations, say, this isn't an excessive number to see an evolutionary change. And you're still looking at 21 years if you're going to look for 100 generations in mice. So fruit flies is getting a bit better. So here we're looking at about 20 days per generation. And fruit flies have been used successfully in the lab to observe evolutionary change. And I'm going to talk about that a bit more later on. And my favorite, bacteria, so they have generation times of about 20 to 60 minutes, obviously depending on the strain, but the typical lab strains that we use have very fast, rapid generation times. And so in these, we're able to observe evolutionary change within a really reasonable time frame. So there have been notable long-term evolutionary studies that have observed evolutionary change. So a great example is Peter and Rosemary Grant, who have dedicated their career to following adaptation of the famous Galapagos finches, which Darwin first noted as really good examples of evolution through natural selection because their beak shape specializes in their food type. So for decades, they have recorded phenotypic and genetic data from these finch species, as well as environmental data such as rainfall and temperature on the Galapagos Islands. And then they've noted key phenotypic changes over time. So between the period of 1970 and 2000, they looked at some key differences between the species Geospiza fortis and Geospiza scandens. And what they saw was that in both species, there was a reduction in body size, but this reduction was more pronounced within G. scandens. They found that there was no difference in beak size over this time in G. fortis, but there was a slight reduction in beak size associated with the G. scandens, and this is perhaps expected given that it has such a dramatic decrease in its body size. But the most notable difference was really in its beak shape between these two species, where they found that G. fortis, its beak changed from a fairly blunt morphology to a more pointed morphology. And even more dramatically, in G. scandens, you found that its beak went from a more pointed morphology to a more blunt morphology. And it was predicted that these changes were driven by changes in their diet, which were caused by climate changes over that 30-year period. So this is a really rich, lovely data set that tells us a lot about the adaptation of these species in a real-world environment. So another long-term evolution study was done by Anthony Long and Molly Burke. So Long established multiple lines of Drosophila in the lab in 1980. And one line was submitted to sustained selection for accelerated development and early fertility, and the other was under no selection. So this is essentially seeing what happens if it just adapts to the lab environment. And they followed these lines for 600 generations. So you might remember that I told you that fruit flies have a generation of about 2 weeks. So 600 generations equates to about 23 years. So this is still a really long experiment. And what they saw was that under no selection, there was a slight increase in time to hatching. So this is from the time of egg laying to larvae emerging. However, in the lines that have been under sustained accelerated selection for accelerated development and early fertility, there was a dramatic decrease in time to hatching. Weight at hatching, again, there was a slight increase when there was no selection and a dramatic decrease when they were under selection for accelerated development. Starvation time in the adults is another thing that they looked at. So we found that when they were not under selection, starvation time was actually, they became more tolerant of starvation. However, when they were under selection for accelerated development, there was a big decrease in their starvation tolerance. And basically, they were pretty poor in every case. So looking at lifespan as well, there was a slight increase in lifespan. When there was no selection, the lab environment is a pretty comfy place to be. But when they're under strong selection for accelerated development and early fertility, you see this massive decline in lifespan. So it's really not a good thing to be under selection for. So both these studies are excellent examples of evolution in action, and they show real-time change of traits in response to selection. But in both cases, these studies took about 30 years to collect and publish the data observing these changes. And given that typically a grant proposal is about 3 to 5 years, it's really hard to get money for a 30-year experiment. And that's because if it doesn't work, firstly, it's 30 years of money, and secondly, it's 30 years of your career. So there has to be another, more accessible way. So in my lab, we use bacteria as a model organism to study evolutionary change. So I'm just using the data from my lab as an example, but there are loads of labs that use microbes to study all kinds of evolutionary questions. So I use the common soil bacteria Pseudomonas fluorescens. It's very easy to work with, it's non-pathogenic. And I look to see how this microbe repairs motility. So if we remove a gene that's essential for motility, how does it repair that? How does it evolve to repair that? So to begin with, we genetically engineer a non-motile strain. So to do this, so this is the wild type. This is how the bacteria exists in a natural setting. It's sort of a rod-shaped bacteria, and it has these long hair-like structures coming at the back, which are like swimming tails. So what we can do is we can genetically engineer this bacteria so that it's unable to make these tail structures anymore. And the consequence of that is that it becomes non-motile. It can't swim on an agar plate. And this agar is like a jelly-like nutrient source that the bacteria can eat and move through. However, what you find is very rapidly, within about 48 hours, mutations that confer this slow swimming phenotype emerge very reliably. And within about 96 hours, you find mutations that confer this fast swimming phenotype emerge. Now, hopefully you've seen here that there's a pretty big difference between the 30-year experiments that we were just looking at. We can see these evolutionary changes happening over the course of a weekend. And then what we can do is we can genome sequence these evolved strains to see which mutations and where these mutations are that are conferring these different swimming phenotypes. So what have we learned from this relatively simple experiment? Well, we've seen that, as I've already said, the swimming speed increases in these two steps. So in this slow swimmer, you get a slight increase in swimming speed. And in this second step, in these fast swimmers, you get a really massive increase in swimming speed. And this is linked to changes within its nitrogen uptake. So what you find is in the slow swimmers, you get a really big increase in the amount of nitrogen that they're bringing into the cell. And in the fast swimmers, there's actually a slight reduction in this nitrogen uptake. And this makes sense when you look at the genetic data, because the mutations that confer this swimming phenotype are actually within the nitrogen regulation pathway. So not where you'd expect them to be, not within the swimming tail pathway. And this slight reduction in nitrogen uptake is to do with the fact that this initial increase in nitrogen uptake actually becomes toxic to the cell. So this is like a compensation for that massive increase here, and it actually makes them more fit. And finally, what we see is there is also a change associated with growth rate. So you see a massive increase in growth rate within these slow swimmers, within the selective environment that they were grown in, and relatively little change after this initial increase within the fast swimmers. So the examples that I've given you are obviously cherry-picked, but hopefully they give you an idea of the power of experimental evolution as just one tool that can help us understand evolutionary processes. And experimental evolution doesn't necessarily have to use bacteria. So there have been labs that use yeast and Daphnia and even cancer cells, and they've all been used really successfully. But they all follow the same definition, and that is that the study of evolutionary change across independent experimental populations as a consequence of conditions imposed by the experimenter. So we must be able to observe evolutionary change, test the reproducibility across replicated populations, and the experimenter needs to have control over the conditions so that we can factor in ecological influences. And the main criteria of the organisms used for experimental studies are that they need to have short generation time so that we can observe evolutionary changes happening within a reasonable time frame. Large population sizes need to be able to be contained easily, and this is to ensure that there's lots of variation within the starting population that selection can act on. And we need to be able to freeze these samples down, and this has two benefits. So firstly, it means that you can keep a frozen fossil record, so you can see the mutations conferring evolutionary change and adaptation over time. But also with bacteria, you can revive these frozen stocks and actually get a direct measurement and quantifiable measurement of the changes in fitness over time from the start to the end of your experiment. And all of this means that you can design your own hypothesis-driven experiments to specifically test the causation of very fundamental evolutionary processes rather than looking for correlations within historical data. So hopefully technology is on our side, and I just wanted to break up the talk a bit by doing a short pop quiz. So this is all anonymous. There's no marks associated with it. It's just pure curiosity. So in a moment, hopefully you'll see these 5 statements appearing on your screen. And then, oh, there they are. And I just want you to select true or false for each one. And I'm just going to give you a few seconds to do this. It shouldn't take long. And then we'll go for the answers straight away afterwards. So the statements are: evolution is random, evolution is well understood, evolution helps us to understand the past, evolution is slow, and evolution is predictable. I'm going to answer 2 because why not? And once enough entries have been submitted, the answers should pop up on our screen.
We've got 13 responses so far, so we'll give it another minute.
That's 10 seconds for the last ones to get in, and then let's just have a look at the results. Okay, I think that's good. I can't see any results yet, though. Oh, there we go. Excellent. Oh, that worked. Thank you so much, Andreas, for organising that. Right, so hopefully you can also see the answers to this. And this is really interesting. I love it. So for most of the questions, you're very divided, and that's sort of the point of them. So especially for this first statement here, it's a 40-60 split. Evolution is random. And also the other one that's clearly split is evolution is unpredictable. Evolution is slow is also pretty controversial. And then the two in the middle, everybody seems to have slightly more sure answers. So the fact that evolution is well understood is assumed to be false, and evolution helps us to understand the past is absolutely true. That's great. That's really, really interesting. And I always love seeing those results. So I'm going to talk over the answers a little bit now. There we go. So the first statement, evolution is random. So for this, I would say this is both true and false. So mutations are random and they are the raw material for evolution, but their survival in a population is not. And this is the process of natural selection. So natural selection will filter out the mutations that confer a fitness benefit. So evolution by natural selection is a complete process, and it requires the randomness of a mutation, but it also requires the efficient selection of these mutations to ensure that the fittest mutations are coming to a fixation within population. So evolution is well understood. Again, I'm going to say true and false. So lucky for me, there's loads of things that we don't know about evolution because it is my career. So I'd be out of a job if we knew everything. But we've learnt loads in the last 150 years since On the Origin of Species was published. And we know lots of things using all sorts of these different methods that I've described. But experimental evolution is quite a young tool that we have, but it's a really important tool that enables us to test these really fundamental evolutionary questions and also investigate how they might influence the evolution of populations in the real world, rather than just looking for trends. Evolution helps us to understand the past. You might be seeing a trend coming up here. So this is true and false. So studying evolution, of course, it helps us to understand historical evolutionary progression. There's loads of stuff that we've learned about the dinosaurs and about the evolution of all the life on our planet. But understanding why things evolve the way they do actually lets us make predictions about the future too. An example that I love about this is vaccine development. So when you get your flu vaccine each year, that vaccine was developed in the previous season. So they can't develop a vaccine that targets the current most prevalent strains. What they have to do is they have to look at emerging strains that are just coming out of the flu populations, and they have to see which of them has a trajectory that's likely to mean that it will be in a high prevalence at the next flu season. And so to do this, they really have to look ahead and make these forward predictions. And sometimes they don't always get it right. Sometimes actually the flu vaccine efficacy goes down because they don't make successful predictions. But a better understanding why things fixate in populations and why certain mutations do well actually means that we can be better at making these predictions. And evolution is slow. So I obviously gave you an example in my lab of where evolution can be incredibly fast over the course of a weekend. And this is because the rate of evolution and evolutionary adaptation depends on many things. So it depends on population size. Bigger populations will evolve more quickly. It depends on generation time. Fast generations evolve more quickly. On the strength of selection. And if you're under really strong selection for a trait, it's gonna reach through the population more quickly. Things like initial variation of the population, stability of the environment, genome size. It's a really complicated thing. It's not an easy question to answer, and there are lots and lots of answers to that question. And evolution is unpredictable. Again, I'm sorry I tricked you. There was no right answers in that pop quiz. So this is both true and false. So the predictability of evolution depends on lots of different factors, and it requires a really deep understanding of the biological system. But it also is really important to consider what scale you're talking about. So are you talking about the phenotype scale, just two things looking the same? Or are you talking about the gene scale? So these two things that look the same, look that way because they both have a mutation in the same gene. Or are you talking about the nucleotide scale? So are you saying that these two things look the same because they have a mutation in the same gene at the same point in that gene? And the finer scale that you get to, the less predictable evolution becomes. So it's important to factor in what scale you're thinking about. But actually, even at the finer scale, sometimes it can be surprisingly predictable. And that's something that I'm going to go into more in this talk and something that my research really focuses on. So a brief whistle-stop history of experimental evolution. I'm going to take a quick drink. So the field of experimental evolution is actually pretty old. So it began with a British Methodist minister and scientist called William Dallinger. or Dallinger, I never know how to pronounce his name, but he was a very early and strong advocate for Darwin. And he was really interested in microscopic life. This was before we could sort of classify organisms at that level. And he was the first to study the complete life cycle of unicellular organisms under the microscope. So between 1880 and 1886, he ran the first controlled evolution experiment. And he did this in a purpose-built incubator, which had 3 chambers, a heating element, and a controlled heat source. And what he did is he filled each of the chambers with pond water that contained unicellular organisms. And then he slowly increased the temperature in the incubator from 15 degrees to 70 degrees over a 7-year period. So it's very slow. At the start of the experiment, the cultures were able to grow at 15 degrees, but were really unhappy at 70 degrees. And by the end of the experiment, the cultures could grow happily at 70 degrees, but they were very unhappy at 15 degrees C. And he took this as evidence of Darwinian adaptation. So unfortunately, for Dallinger and for us actually, this incubator was accidentally destroyed in 1886. And since then, this type of research was actually abandoned for a long time, for about 100 years. And that is until this chap turned up. So this is American scientist Richard Lenski, and he began a long-term evolution experiment in 1988 that revived the field of experimental evolution in a really big way. So Lenski established 12 independent cultures of bacteria E. coli. He grew them in a minimal nutrient media, so there's not a lot of resources to go around, at 37 degrees C. And every day he transferred 1% of the cultures into fresh medium. And this equaled about 6.7 generations per day. So Lenski also periodically stored these samples at minus 80 degrees C. And this left a really detailed fossil record of adaptation and a really nice genetic record to go back and look at. So he set up these 12 independent populations in 1988. And they're still running today. And I checked about an hour before this talk, and they were up to 76,272.2 generations of E. coli, which is mind-blowing. And if we're assuming about 20 years per generation for a human, this is equivalent to about 1.5 million years of human evolution. It's really staggering. It's a really incredible experiment, and yet so beautifully simple. So what have we learnt from this experiment so far? So this is really just skimming off the surface. There's loads that we've learned from this so far, but looking very generally, we've kind of got a really nice overview of what adaptation looks like over a really long time period. So firstly, looking at the fitness trajectory at the first 2,000 generations. So fitness here is a measurement of the ability of this bacteria to grow and to reproduce within its environment. And it's all relative to where they started off. So they start off with a fitness of 1, and anything that goes higher than 1 means that they're getting better at dividing and growing in their environment, and anything less than 1 means they're getting less fit, they're getting poorer. And this is over 2,000 generations we're looking for at the beginning. And what this shows is that after 2,000 generations, we're looking at about a 30% increase in fitness. But the trajectory of this obviously isn't smooth. You're looking at these stepwise increases of about 10% each, 3 big leaps. And what this shows is that each of these steps is dominated by a few mutations, but with really large effects. So they really have big fitness benefits associated with them. So other mutations do occur as well, but they're outcompeted by the most beneficial ones. So when these clones from these populations were sequenced, genome sequenced, they found that there were 5 mutations that were shown to be beneficial within these bacteria. And the fact that there were 5 were interesting because it's not that each one of these steps is determined by one single mutation. In some cases, you needed multiple mutations to occur at the same time in order to allow this big fitness leap. So what about if we look over a longer term, a longer time frame? So again, we're looking at fitness here on the y-axis, starting at 1. This is where you start your experiment, and looking over time up to 50,000 generations. And here you can see the trajectory is much smoother. But you've obviously got this massive increase in fitness at the start of the experiment that we saw over here. But then you see that it starts to level off. So here at 50,000 generations, you're still looking at about a 70% increase in fitness. So they've had a really massive fitness advantage over this time. even they've given so much time, they're still increasing in fitness. But does this slowdown imply that the bacteria have or will hit some sort of limit on their fitness? So the way that they've addressed this question is by fitting a lot of mathematical models to their data and just seeing which one fits the best. And so far, they've found that the power law model is the best fit. And this generates predictions far into the future. And what it suggests is that it doesn't actually reach a maximum. However, what it also shows is that after about 2.5 billion generations, the growth rates start to become biologically quite implausible. So it could be that that's about the limit, but that's still a really, really long way to go. So it'll be really interesting to see how long this experiment continues, and why would they stop it? I don't quite know what would make them stop, but it's been interesting actually. So this pandemic has been the longest pause within this long-term evolution experiment, which has been running, even over Christmas Day, every other day since then. So they did shut down the lab and pause the experiment over the pandemic, but it's back up and running again. So these simple experimental evolution studies have provided a foundation which we can begin to build a better understanding of evolution. But can we then apply this knowledge to make better predictions about evolutionary outcomes in real evolving populations? And to what benefit? Why does it matter? Well, there are loads of really important applications to this knowledge. Things like trying to preempt the emergence of antibiotic resistance bacteria, which we know is a huge problem in multi-resistant bacteria. So what if you could preempt which mutations are likely to emerge under certain treatment regimes and adapt your treatment regime to try and stop these mutations emerging in their tracks? The same is true for cancer treatments. So you often see drug resistance evolving in cancer cells and in tumours. So what if you could preempt which mutations are likely to arise and design your treatment protocol to make sure that you take into account these new resistant lines that are likely to emerge? And timely as it is, it's really important to understanding the emergence of pathogens and viruses and understanding what happens in a novel host. So we've all heard a lot about these new variants that have emerged in coronavirus and specifically on the spike proteins, which is the bit that's going to be under the strongest selection. So we're always playing catch up. We have to now redesign our vaccines in mind of these new variants. But what if we could predict these variants before they emerge? That would obviously be a massive benefit in terms of a vaccination program. So mutation is inherently random and therefore full predictive power is highly unlikely. Okay, but a better predictive power is certainly possible. And that's through understanding underlying factors that are likely to bias which mutations are likely to reach high frequencies. So what do we need to know in order to predict evolution? So imagine that within this bag, there are a number of different mutations, and these mutations confer a certain phenotype, okay? So firstly, we need to know how many different mutations there are that confer this phenotype, how many different colors, how many different counters are in this bag. Then we need to know the probability that each of these mutations is going to arise given the type of mutation and the genetic background. So it could be that some of these mutations just aren't viable in certain genetic backgrounds. So as much as they would produce a phenotype, they are incredibly maybe unfit in that host. And so you wouldn't expect them to reach any sort of fixation. And the other thing you need to consider is the likelihood that each mutation will reach fixation in a population. And this is going to depend on two things. So this hand here, you can think of as selection, is the selector. So it could be that the selector has a favorite color, and so they're more likely to pick one of these, say, orange counters in this bag. So that's what you can think of as sort of positive selection. Or it could be that certain mutations are overrepresented and they occur more frequently, and therefore the selector is more likely to pick them. So this is natural selection, and this here is actually a form of bias. So within my lab, we use the common soil bacterium Pseudomonas fluorescens. It's nice to work with, it's non-pathogenic, and it's fairly, it does as it's told most of the time in genetics in the lab. And it was isolated originally from the leaf of a sugar beet plant in Oxford in 1989. And we used this bacteria as a model organism to study the evolution of genes and gene regulation. But we've also been using this system to look at the predictability of evolution. And this is from another direction. So what if you have a system that shows really predictable evolution? So can we identify what might be driving that process? So this is an example of starting with the most simple case. So before we can understand how to predict evolution in a system that seems unpredictable because of its complexity, we can instead ask the question, okay, when evolution is remarkably predictable, can we identify the drivers at play? So what we've done, and I've mentioned this before, is we've genetically engineered this bacteria to be non-motile. So when you put it on an agar plate, it doesn't move anywhere. And we can change the strength of selection by looking at this, how it evolves motility under high and low nutrient media. So LB is high nutrient media and M9 is low nutrient media. If you leave these bacteria in the middle of an agar plate, very reliably, they will evolve a swimming phenotype very quickly. And then we can genome sequence these swimming mutants to understand what are the mutations underlying this phenotypic change. So we've now repeated this many times, and we started to notice something quite interesting. And what we noticed, so if you imagine each of these colors as a different colored counter in that bag that I was showing you earlier, we noticed that there were very few different colors. There are very few different mutations in the evolved bacteria. So this y-axis here is showing the proportions of all the different mutants that we picked, the proportion that carried each of these mutations in a high nutrient media and in a low nutrient media. And overwhelmingly, this mutation was the most common. So I'll just quickly explain the nomenclature. It's not that important, but these first 4 letters at the beginning, this is the gene NTRB, and this is saying that there was a mutation from a base A to a base C at site 289. So A289C is what we've been calling it. So both these mutations, and this means a deletion. So this just means that these bases, 406 to 417, were deleted in the NTRB gene. So massively, this NTRB A289C mutation was overrepresented in our population. So we wanted to ask the question, what could be driving this predictable parallel evolution at such an extreme level? So this is very rare to see. And again, as I mentioned previously, the scale is important. So frequently we see parallel evolution or repeatable evolution at the phenotype scale, but it's much rarer to see it at the nucleotide scale to this degree. So we had 3 hypotheses as to what might be going on. Either that there were no mutational routes available, so it's very constrained as to what it could do. Perhaps natural selection was acting really efficiently, and this overrepresented mutation is the fittest by far. Or it could be that there's some kind of bias involved, and that means that the common mutation is just more likely to emerge than any other. So to start off with, we tested our first hypothesis, which is that evolution was incredibly constrained and there were no other viable mutational routes available. So to do this, we just repeated it a lot. So this is over 100 times we repeated this experiment, and in the high nutrient media, a low nutrient media, and also this is a low nutrient media with just a single amino acid added. And the reason we looked across these environments is because we wanted to understand whether selection was playing a role in which mutations emerged. And very much we found it wasn't. So this again is looking at the proportion of mutants that we found in all these different environments. And the same story, this NTRB A289C mutation was massively overrepresented in everything that we looked at, all our populations. But we did find some other alternatives. So what this means is that we can actually reject this first hypothesis because all of these were swimming motility mutants. So this says that there are other viable routes, but we're just not seeing them at high frequencies in the population. So our second hypothesis is that natural selection was just doing a really good job. So what this says is that the reason that there is such a high representation of this NTRB A289C mutant is because it was by far the fittest. So to check this, we measured the growth rate of this mutant and then compared it to the growth rate of all these other mutants here. So this is the fitness compared to this common mutant. And what we saw is that for 3 out of the 4 mutations that we'd identified, there was actually no statistically difference within their fitness compared to this common mutant. So they were comparable in terms of their fitness. The only one that we can actually rule out as being far less fit is this mutation in a separate gene, GLN-K. So selection is doing a great job at removing that. That's fine. We wouldn't expect to see that at very high frequencies within our experiments. The other ones we can sort of rule out are these, or we can be suspicious of at least, are these 2 mutations here, which are these 12 base pair deletions. And the reason we'd expect to see them at lower frequencies is just a probability argument. So in that case, you're changing 12 base pairs at one time versus the other two mutations where you're just changing one base pair at a time. So the probability tells us that we're more likely to successfully change one than we are to change 12 at a time. So you might expect these two to occur at a lower frequency also than our experiments. But this one was very tricky to explain. So this is also an A to C mutation. So it was an A and it mutated to a C at site 689 in the NTRB gene. So it's within the same gene. It's the same type of nucleotide change, and it's only just a bit downstream of this other mutation. So if everything was equal, we would expect to see this mutant at higher frequency, given the fact that it has a comparable fitness. So this means that we can also reject our second hypothesis. So selection, if selection was the only thing at play, we would expect to see the NTRB A689C mutant at a higher frequency. So this leaves us with our third hypothesis, that there's some sort of bias going on, that the common mutation is there because it's just more likely to emerge than any other. So how can we go about testing this in a meaningful experimental system? And this was quite tricky. So I'm just going to give you a really quick basic 101 on bacterial replication. So in bacteria, they don't have a nucleus. The genetic material just exists within the cytoplasm as a sort of tangled mess. And were you able to lay it out flat, you see they have a single circular chromosome. And this chromosome, it stores all the DNA that's used to decode, is decoded to create these proteins. And proteins are actually the important thing. Proteins are the things that do things in your body. So DNA is actually just an information storage mechanism. And it's the proteins that selection sees and can actually act on. So it's because it's proteins that will change certain traits. So you have certain enzymes that are whizzing around this chromosome. You have DNA polymerase, which is responsible for copying this DNA. So if the cell's going to divide, it copies DNA to more DNA. You've got RNA polymerase, which is copying DNA to RNA that can then go off to interact with the ribosome here, that then translates this mRNA into an amino acid chain. And the third thing I just quickly need to tell you is how this DNA code is read. So there are 4 bases, A, T, C, G, and this code is read in triplicate, so in basically 3-letter words. And because there are 4 bases and 3-letter words, this means that there's 64 combinations that can come out of this. But there are only 21 amino acids. And so what that means is there's a certain amount of redundancy. So some 3-letter codes will code for the same amino acid. So for example, here you've got TTC, which codes for a lysine. And if this third base position were to mutate to a T, you still code for lysine. So TTT also codes for lysine. And this is known as a silent mutation. And the reason it's called silent is because it doesn't cause a change in the protein. And if it doesn't cause a change in the protein, it's considered silent because selection won't see it. However, other mutations can be coding changes. So in this case, if you change the second base position here to a C, to TCC, you end up with amino acid arginine. And this might lead to changes within the protein structure that can lead to differences in fitness. So because of this reason, because these mutations are considered silent, they're kind of mostly ignored from adaptive processes as sort of being important. But actually, we have evidence now to show that they can be incredibly important in determining the evolutionary outcome or evolutionary trajectories of these evolving populations. So the reason I showed this is not really for you to understand everything, but to show that there are lots of different things that can make mutation rate vary across a genome. So it can come from collisions between polymerases and from high repeat sequences, or even just where you are on the genome. It's not as easy to say that there's one single mutation rate that exists around the whole genome. So we tried our best to sort of eliminate as many of these options as we could. And there was one thing in particular that really piqued our interest. So this is number 6 here, this hairpin structure. And what can happen with these hairpin structures is that these polymerases can come along and sort of bump into them and stumble and trip. And because they trip into them, they can cause a higher mutation rate at the base of these hairpin structures. So I just want to sort of visualize that a bit for you. So we know that DNA, when it exists in its double helix structure, they pair together, so A with T and C with G. However, when a polymerase comes along, this structure needs to actually separate, so they become single-stranded, they move apart, and this allows the polymerase to come along and copy a single strand, okay? But what can happen by accident is you can get these repeat sequences. So here you can see ATC, GAT, CGAT, and here, again, ATC, GAT, CGAT. And these actually complement each other. So once these strands separate, they're actually allowed to come together and form their complementary pair in what's known as a secondary structure. And this can cause problems, as I said, for a polymerase traveling down the DNA, because it can bump into this structure that it doesn't expect to be there, and this can cause an increase in mutation rate at the base of these hairpin structures. So we saw a tantalizing suggestion of a hairpin structure at the site of this highly mutable site within our bacteria, but it wasn't enough evidence. So we needed to think of a clever way to test this experimentally as to whether this is in fact the cause of our bias that we're seeing in our bacteria. So luckily at the time, another PhD student, Louise, was working with another strain of Pseudomonas fluorescens. These are both the same species. So far I've been telling you about this strain, SBW25, isolated in Oxford. This strain, PF01, also Pseudomonas fluorescens, was isolated in Massachusetts in the US a couple of years later. And we've known from previous work that we've done that if you knock out the gene that allows the swimming flagella tails, again, you get this non-motile phenotype, and they will re-evolve this swimming phenotype very quickly. So we knew the same process was at play. So that's why we were looking at it within our lab. So we were trying to understand the mutational diversity within this strain. And it's incredibly different. So here, this PF02X is just the code name that we have for our non-motile PF01 mutant. This again is looking at the proportion of mutant lines on the x-axis in a high nutrient media and in a low nutrient media. And if you imagine these are all our coloured counters within that bag, it's a far more colourful place. There's just loads and loads of variation. It's still happening within the same genes that we would expect, so this NTRB gene, and these are also genes associated with the nitrogen regulation pathway. So these are all within the same pathway, but they're just massively diverse. So we thought, okay, this is super cool. But the thing is with speciation in bacteria, it's really hard to classify what's a species. And that's because bacteria don't sort of behave as sort of sexual organisms. We can't look for reproductive isolation or anything like that. So what we needed to do is have a look at, especially this site, this NTRB gene, which you knew to be a site of high repeatability within our other strain, and see, well, how similar are they really? And so this here shows how similar they are within the DNA. And this shows they're about 89% similar. And this is the base that we frequently see mutating within our original strain I was telling you about, or SBW25 strain. And this is the amino acid alignment here. So this just shows the two amino acid sequences aligned against each other. And again, we're looking at about 96% similarity. So these are really, really similar. And this is the site that we frequently see mutated in our other strain. But if you look, so where you see a gap, it basically means that these two sequences don't quite line up. There's a difference between these two strains. So we're looking at the amino acid level, there's no real differences around this frequently mutated site. But looking at the DNA nucleotide level, you can see there are these gaps and there are these differences. So our theory was that these differences were actually disrupting that hairpin structure, and meaning that it couldn't form, and therefore that was the difference into when you were seeing this highly predictable evolution versus this very unpredictable evolution. So what we then thought, okay, so what we'll do is we'll engineer the bacteria so that the strain that shows very predictable evolution, we'll engineer it so that it's more, so we'll disrupt this structure, so we make it more like PF01, but in doing so, we won't change the protein structure. So selection should see it the same. The protein that comes out of this DNA sequence should be exactly the same. All we're doing is disrupting the secondary structure in the DNA. And what we found was it was better than we hoped, really. So this again is showing that same graph I showed you before, what happens in high and low nutrient media. We find that this A289C mutation massively dominates. But when you do have this silent, SM stands for silent mutant, when you have the silent mutant, you see a massive array of diversity and all sorts of mutations we've never seen before. And importantly, we've never seen this A289C mutation within this silent mutant. So we seem to be able to successfully break this mutational hotspot by just introducing 6 silent mutations within this genetic background. And conversely, if you look at what happens within the PF01 strain, here you have this glorious diverse array of mutations here. And when you genetically engineer this strain to have just 6 silent mutations that make it more like AR2, suddenly you see that this NTRB A289C mutant dominates, something that we've never seen emerge naturally in this strain, with just a few exceptions. So in this case, we seem to have successfully built this mutational hotspot through just 6 anomalous changes. So we've gone from something that evolves very predictably to very unpredictably, and conversely, we've been able to actually construct, to engineer a mutant that goes from something that evolves very unpredictably to something that evolves very predictably. So what does this tell us? Well, it really does challenge our understanding of the role that these silent mutations play in adaptation. So it might be that even though selection can't see it in the sense of seeing it in the protein, the mutations that are revealed to selection are actually really different, even though the mutations within the DNA don't actually do any protein change. And it raises questions about how often mutation bias might actually be biasing adaptive outcomes. So how often does this happen? Is this just a freak of nature, or is it actually playing a role in adaptive evolution all the time? And it highlights the role of underlying molecular factors in driving predictable evolutionary events. So it really says that we need to understand the fine details of these evolutionary processes if we ever hope to be able to have any sort of meaningful forecasting ability for evolution. So I want to go back and think for a moment about the questions that I posed at the start of this talk. So what are the factors that determine evolutionary outcomes? Well, there are many, and they are complicated. So there's lots of moving parts. And of course, selection plays an incredibly strong role, but there are other factors at play that we need to consider and we need to understand. So are these factors ever predictable? There's lots and lots of randomness in evolution, and it seems like a losing game to even think that we could ever predict it. However, there are things that can constrain the possible outcomes, which make predictions easier. But we don't yet understand enough about what these factors are and how often they're acting, so that we can recognize where they might be at play. And finally, how could we explore these questions experimentally? Well, hopefully I've tried my best to tell you that experimental evolution gives us a really excellent basic toolkit that allows us to replay the tape of life. And these allows us to understand questions just like this. And it's important to start simple, to understand the most simple cases before we can start to move up in our complexity. And with the rapid advancements in genetic technologies, we're able to understand these processes at a molecular scale, and this is beginning to reveal really surprising discoveries. So it's really important that I acknowledge my PhD students that actually did all the work. So James Horton and Louise Flanagan, they collected all the data in the parallel evolution studies that I talked about. Matthew and Josie are also 2 excellent PhD students in my lab, and Aidan Pearce is our awesome research assistant. These are other academics that have been really influential in the progression of this work. Funders are BBSRC and the Royal Society, and if you're interested in how this story progresses, you can follow me on Twitter. And thank you so much for your attention, and I'll happily take any questions.
Well, thank you very much for that, Tiffany. So we're now going to move into the Q&A part of our evening, and I'm going to ask Catherine Laycock to ask a question, but I suspect it's not actually Catherine Laycock. Is that right?
No, it isn't. It's Jolyon Laycock. We're both members of Brisley, and we're just using Catherine's computer.
Well, good evening, Jolly. Would you like to go ahead and ask your question?
Well, yes, I was interested. Yes, thank you. A fascinating talk. Thank you very much. I was interested in the idea that, for instance, what's the thing called? The Pseudomonas florensis, and also the generation of E. coli, something like 76,000 generations within a reasonable experimental time period. Is it possible to conceive that at the end of a program like that, you might be looking at the arising of new species? Is that a possibility?
It's a great question. The thing is about these experimental setup, especially about the Lenski setup, is it's designed to be incredibly simple. So there aren't really any different niches for these bacteria to move into. There aren't many nutrients actually for them to access. So it's like a minimal nutrient. So what you see, if you look at the sort of mutational profile over time, is that you see these mutations coming in and sort of sweeping through to fixation. So you'll find that one mutation emerges that leads to a fitness benefit, and it sweeps through the population, and that will happen again and again. So that mutationally, we're at a very different place, but in terms of what it can do in terms of bacteria, it's probably not diverged enough to actually call it a new species. But then I did touch on this, that speciation in bacteria is really, really difficult to define. How do you define a species in something that is reproduces asexually? Because our normal understanding of what a different species is reproductive isolation. So when no two organisms can't come together to produce a viable offspring anymore. And that can't happen, obviously, in bacteria. So we have to look for these sort of arbitrary thresholds as to what makes one bacteria different than the other. And so that's why within even our fluorescence, we had to go back and really compare the genomes of what we were looking at to say, okay, they're the same species, but that doesn't actually mean that they're that similar. So there's some, what we call within bacteria, you have the core genome. So these are the genes that are shared by all the bacteria. And then you have an accessory genome, which sort of describes all these other genes and these other mutations that can be present. And there can actually be massive differences between them. So you can get quite small core genomes and these huge accessory genomes. So in answer to your question, I don't think we haven't quite got speciation yet, although there was a very excellent, a really interesting population that basically was able to use citrate as a nitrogen source. Now, normally E. coli can't do this. So this was a really novel, really exciting adaptation. And it's possible that if we were to find that strain within the wild, we might think of it as something different than E. coli, because it actually behaves quite differently because it can utilize this other resource. But as a very long-winded way to say that speciation is really difficult to classify in bacteria. So I'm going to say no, it's not, but that's not necessarily an easy answer.
Thank you. Thank you.
Thank you.
Thank you.
Thanks.
Okay, we have another question from someone with a very interesting name here. They're called iPad. I wonder if I could ask iPad to ask its question. Hello there. Good evening.
Hello. Thanks very much. Thank you for a very interesting talk. I was wondering about whether there was general sort of selection pressure for things to actually be more likely to evolve. I mean, an obvious one would be if you've got things competing, one of which has a shorter time between generations, or the other possibility would be one is, if you like, slightly less stable and therefore more likely to mutate, and therefore going down the generations, there'll be different generations, whereas each one is a bit more like its parent. Whether there was actually either of those represented a kind of selection pressure that.
So whether sort of a high mutation rate would actually be beneficial under certain environments.
That's right, or even a shorter time between generations, just as
yes,
it can sort of leap ahead of its competitors.
Yes, definitely true in terms of shorter generation times. That can absolutely be beneficial in lots of scenarios. As long, I mean, I guess a really interesting example of that, what may be interesting, is sort of in cancer, right? So what happens in cancer is that you have a cell that breaks its multicellular programming so that it divides at a faster rate than it essentially should for the good of the organism, right? So it essentially kind of acts selfishly as an individual cell rather than acting as a group. And so especially if you're in sort of a group environment, finding a way to outcompete that group. So if you're sort of constrained by that group, it can lead to a massive advantage, at least in the short term. Obviously, cancer isn't a long-term goal, but in terms of fitness in that moment, you get an advantage over your competitors. And in terms of mutation, again, that really depends on whether or not we're interested, so whether kind of on your environment. So if you're in a very harsh environment, an incredibly stressful environment that basically is do or die, you either find a fix or you're going to die. In that case, you find with bacteria, a natural stress response is to increase their mutation rate. So that's what they'll do under stress. And that's just to try and basically throw everything at it and just see if a mutation that confers a way out of this stressful situation just pops up. So it can be really advantageous. But the trouble is with having that on all the time is that most mutations will have a negative fitness effects. Most mutations will be deleterious because they'll change, especially if their coding changes, they'll change the protein in such a way that means that it won't function properly. Because selection has already sort of modified the genome to be pretty good at what it does. So most mutations won't be helpful. So it's only really under these extreme stressful environments that high mutation rate is beneficial. But it's great. It's kind of amazing to see that bacteria have this as an adaptation themselves when it gets stressful, just to become much more mutable and to find the right mutation quicker.
Right, thank you very much.
Thank you.
Perhaps I could ask about what you've just said, Tiffany. Mm-hmm. It seems to me very difficult to think of a way in which the adaptation of having a higher mutation rate could evolve. I mean, the way that you explained this to us was by supposing that it's advantageous under very stressful conditions. Mm-hmm. But that would imply that it's probably not advantageous the rest of the time.
Exactly.
And it seems very difficult to understand how a complex adaptation like this could arise.
Yeah, that's a great question, and it's not one that I probably have a very good answer for. But I mean, I think so. These general stress responses actually. So these bacteria kind of they basically upregulate their entire genome. It's kind of what they do. They just upregulate everything. And one of the things that means is that their enzymes that are usually used for sort of checking the DNA just go a bit haywire. So it's probably, I mean, how that evolves is a really, really tricky question. And I don't know whether that's maybe a consequence of actually just increasing, I guess within those conditions, you're going to be upregulating lots of genes associated with sort of, I don't know, so if you're in a very starvation environment, you're going to want to upregulate all the genes that pull nutrients into the cell, or if you're in a very toxic environment, you're going to want to upregulate a lot of your genome. So I guess it's associated with these massive changes within whenever you're exposed to these really harsh environments, you have to almost completely change your gene expression makeup. And I think it could just be that this sort of high mutation rate is an artifact of that. Whether or not it's an evolved response, it's really hard to say, and it's not something that I know the answer to. But I'm probably going to go away and try and read a bit more about it now that you've said it.
Yeah. I suppose that one possible explanation here is that the adaptation, the way that we're looking at this adaptation, which apparently causes a higher mutation rate, actually has some other functions.
Yes.
Under less stringent circumstances.
Yeah, it's really important to remember that, say, not everything is sort of adapted to suit its cause. Actually, there's lots of things that are sort of accidental, that they happen as a consequence of something else that can sort of provide an accidental benefit in environment. So yeah, it's important to remember that not everything has been specifically selected for its purpose.
Yeah. Okay, we're not getting too many questions here, so I'm going to put out an appeal for you to ask your questions here. But I'm going to ask another one just to keep things going for a bit. It seems to me that the design of the long-term experimental evolution experiment is kind of unlike the real world, in the sense that what you have is a step change to a new environment, which then remains absolutely constant. Yeah. I mean, it seems to me quite likely that evolution ordinarily involves the proliferation of an organism which is actually going to alter its environment simply by living in it. And I wondered if you had something to say about that.
Sorry, say your question again. Sorry.
Well, I guess that I'm talking about what's sometimes technically called niche construction.
Yeah. Okay.
Which is that ordinarily, I think that probably most organisms actually modify the environment that they live in. And so if that's the case, I was wondering whether evolution is constantly churning, because as you evolve to modify your environment, the environment isn't necessarily going to change in the way that continues to benefit the organism, the mutation that caused it to change in that way.
Yeah, I mean, that's true. I mean, so yeah, the long-term experiment, I see what you're saying. So that's sort of, it's very artificial in the fact that it sort of replenishes this environment every day. So they're sort of always encountering the same environment. There's been some really nice studies that have been done with sort of looking at the competition and the sort of the relationships that can evolve with sort of more than one species within this environment. And what actually you can find is that quite quickly they will find niches even in very simple environments, and it's such like mutualisms can evolve quite quickly, where sort of the waste product of one bacteria can actually become the nutrient source of another, and in that ways they can establish these communities. But yeah, sort of so when you put these organisms within these communities, there's actually a classic example with Pseudomonas fluorescens, where what they've done is they've just put SBW25 in a static broth, so that it's basically in some liquid, and they let it grow, and quickly they establish these niches where one has a mutation that means it can sit at the surface, one has a mutation that it sits at the bottom, and one has a mutation that it kind of swims through the middle. So yeah, if you allow these complexities to evolve, they ultimately will, and they will shape their own environment, like you say, and especially this becomes more and more important the more species that you add in. So these complexities between organisms become really important. And we're only really getting close to these sort of multi-species interactions now. It's taken us a long time to understand the basics. So we're getting to a point now where we're exploring these ideas. But yeah, absolutely, it's a factor that an organism shapes its own environment. And one of the other things actually they've started doing is within soil microcosms as well. So they're growing these bacteria in soils to try and emulate a more natural environment. And yeah, you quickly find that they establish these niches and they diversify much more quickly. So this is very artificial to have these sort of sweeps that I was talking about. It's due to the sort of simplicity of the environment that sort of maintains that trajectory.
Oh, that's really quite a nice idea, that it might actually be an inevitable outcome of population level evolution, that you generate genetic diversity within it, which is perilly close to something that we used to be told not even to think about, which is group selection.
Oh, right, yes.
Anyway, we've got some more questions come up now. David Cameron has a question. Would you like to unmute and unveil yourself, David, so we can all see you, if indeed you are David Cameron, in fact?
Yes, indeed. Oh.
Hello, David. Would you go ahead and ask your question, please?
Okay, thank you. Thank you for the talk. Just a question. You mentioned that environmental stress increases the mutation rate in bacteria. Is this also true in other organisms, such as animals, for example?
It's not. So I think for the reason that happens in bacteria is because they have this really fast replication rate. So I think without that, and also asexual reproduction. So that's really important here, that any mutations that occur within that background are guaranteed to go on to any daughter cells. So having that asexual reproduction is important. I don't know of it happening in any more complex multicellular organisms. I hope I'm right in saying that, but I haven't heard of that happening. No, I think it's unique to unicellular organisms.
Thank you.
Thanks.
There are cases where the recombination rate can be modulated in stressful conditions. So we're not talking about new mutations there. We're talking about just remixing them up. Okay, I've got another question here from BRLSI Zoom, which I strongly suspect to be Andreas Vasmuth. Is that right?
Absolutely right, but it's not much of a question. It's very much more of a sort of observation, and that is that the bidirectional interplay between the organism and the environment is particularly interesting, especially at times of instability in both the organism or the environment. And I just wondered, to what extent have you been able to vary the environment in what you've been doing?
So far, we are keeping it really simple. So there is something interesting, actually. So within the strain that shows the very variable mutation profile, there seems to be an effective environment on that. So that's why we've sort of been looking at these low and these high nutrient environments, especially because we understand that the mutations within those backgrounds are messing about basically with their nitrogen uptake. So we've been sort of altering the nitrogen environment to sort of make those mutations more or less costly. And so that we can see in the one where it has lots of variation that it favors mutations in certain genes compared to others, which is something that we don't see in the other strain. And so that's really interesting in sort of a terms of, so what does this hotspot mean in terms of sort of evolutionary consequences? So if these two bacteria were sort of competing in environment, one of them had the sort of defined predictable route and the other one had sort of the more diverse route, which one would win out and which one would actually sort of be able to occupy more space? So that's a question that we're getting at. And we've talked about things like making the agar plate sort of patchy, so changing how where the nutrients are distributed, or changing it over time, so perhaps switching them between a high and a low nutrient. These are all thought experiments at the moment, so we're thinking about it, but at the moment we're sort of keeping things quite simple, but there is the hopes to sort of include some of these factors in the future experiments.
Good, thank you very much.
Thank you.
The next question on my list here is from Tony Symes. Would you like to ask your question, Tony?
Yes, I'm not a biologist, so but I understand that genes can be switched off by a process called methylation, and that means that an organism could have genes kind of in reserve and only just make use of them in later generations. Does that play a part in your...
Well, yeah, so I mean, in bacteria, methylation doesn't occur, but you do get gene regulation. So absolutely, they can change which genes are switched on or off. That's through the sort of transcription profiles, which is something that we're actually really, really interested in our lab. Something that we're sort of looking at is how do you evolve the capacity to be able to regulate your genes. So what we've seen is that it's within our motile mutant, this is sort of going back to a previous experiment. So when we break the motility and we allow it to re-evolve, the way that it does that is it kind of borrows a regulator from a different gene and it uses it, plugs it into this sort of gap that we've created by deleting this other gene. But the consequence of that is that it loses its ability to switch on or off either of these networks. So it's constantly churning out its flagella and it's constantly drawing up nitrogen from the environment. So it becomes clear that these the capacity to be able to sort of modulate your gene expression based on the environment is really, really important. And that's something that sort of, we think it's actually really, really important for the way that these networks, so these gene networks evolve. So they evolve and they occur in networks because it's much more efficient to be able to switch on or off a group of genes rather than each one individually. But we think that an important factor for the way that they evolve and the structures that evolve is how switched on they are or how switched off they are at any one point. So those that are consistently on are much more likely to be co-opted to sort of recover function. So whether or not they're on and off might have a really important factor in terms of how evolvable those genes are in terms of a network. So that's really interesting. And yeah, I mean, I think in higher organisms, I don't know if I can really say much about the process of methylation. I mean, it's interesting to sort of think about this idea of, so what you can get with these mutations is you can get these degradation of certain genes. They become pseudo genes. That basically means they become non-functional, but because they kind of are a relic of a gene, there's evidence to suggest that they can actually evolve to become functional again and sort of recover or be co-opted to new roles. So having these silent genes or these sort of switched off genes within your genome has sort of been shown to be beneficial for sort of evolutionary processes, because it's kind of a material that they can use to sort of evolve, because it's almost a ready-made gene. It's not starting from scratch, it's sort of almost there, and they can modify it a small amount to sort of make it functional. So they are important in determining sort of the rate of evolution. I
have a question. May I ask?
Tony, please go ahead and ask your question. Why don't you switch on your camera so that we can enjoy looking at you?
I tell you why, there's a simple answer to that. It's because I can't. I don't know.
Okay, but go ahead, Tony, and ask your question.
Yeah. I'd like to ask you about where this is going to lead, where this is leading to. Is it the hope and the ambition that if you get clarity in this field, you will, well, I want to know what your hopes are, where this is going to lead to. That's my simple question. Really, I'm asking, do you think it's feasible that we will be able to predict the evolutionary outcomes in bacteria and in higher animals?
I mean, as I said, I don't think full predictive power is possible because of this element of randomness that is unpredictable. But I mean, one of the next things that we're trying to do from this, so we've discovered this sort of really interesting case of a direct cause of sort of parallel evolution at the molecular level. But now, what we don't know is this a real fluke, or is this actually something that is missed a lot in these sort of where you see instances of parallel evolution? Are these underlying biases that really exist? So something that we want to move forward to do is sort of try and look for these instances of where you see very high mutation rates in the genome, and just to see, can we see any common signatures that might indicate underlying reasons for the bias that then we can sort of search for more freely in genomes and say, okay, so what we think is when we see a high mutation rate, we see these inverted repeat sequences quite commonly. So, okay, so now let's go through lots and lots of genomes and look for these inverted repeats and see, well, do we see a high mutation rate? And if we do, then what that means is, say for things like looking at a coronavirus, if you see these inverted repeats there, then you can make a plausible prediction that, okay, well, this is likely to be a site of high mutation. And so we could then use software to sort of predict, well, what would mutation at this site do to the virus? And so it's sort of having that power to sort of to look at how these changes might be sort of actually acting in the real world, and sort of look for these signatures within genomes that would allow us to make these educated predictions out in the real world. I think that's our hope.
Thank you very much, Tiffany.
Thank you.
Okay, I'm going to ask one last question. I guess that I'm going to take advantage of the fact that I've got the floor to ask a last question. And I was really very interested by your finding that these DNA hairpins were important in determining the mutation rate. Do DNA hairpins occur equally often in all bacterial groups, and are there also DNA hairpins that occur frequently in the genomes of eukaryotes?
Yeah, I mean, DNA hairpins are really just a freak accident. I think there is evidence to suggest that they are in areas where you don't want high mutation rates. There is selection to remove these inverted sequences from the genome. So ultimately, they tend to be under selection not to be there. But in an area where they're unlikely to cause any harm, then I think they can accidentally occur. So you have to remember that within the bacteria that we're using quite a weird genetically engineered mutant. So it's not something you'd ever see in nature, to have this sort of non-motile mutant hanging around without an entire gene that then has to evolve a way to fix it. So normally this hairpin structure wouldn't be seen by selection. So I think it's basically not been selected against, and so it's been allowed to persist within the genome. But as to whether or not, you know, these hairpins are, how prevalent they are, and the role that they're currently playing in sort of adaptive evolution, that's a real unknown, and that's something that, you know, we really want to move forward with the project and try and
well, it's actually a commonplace of evolutionary biology that there are some lineages that evolve very quickly and others that don't. And I guess it would be really quite interesting to look at the frequency of hairpins in those different lineages.
Yeah, definitely. And I'm also really interested, sort of thinking back to the Lenski experiment, so when you're looking over the really long-term scales, so what are the differences in terms of the drivers of adaptation at the very early stages, which you expect that to be very strong selection, versus the later stages where there's going to be weaker selection in action? Is that where you see these other sort of biases coming in or being more prevalent? That's an open question.
Terrific. Well, thank you very much, Tiffany. I think that we've all had a very interesting evening, and we've learned how actually doing experiments can help us to understand something which Darwin himself couldn't even have imagined. It is the mystery of mysteries how adaptation actually takes place, and to know that there's a way of actually looking for the mechanism in a test tube is really astonishing. And we wish you great success in applying these ideas to real life situations. I was very interested in Tony's question about where you were going with all of this. Maybe you'll come back in 20 years and tell us where you've got. So,
well, it's all thanks to a really great lab group, to be honest. So it's not, I'm not taking credit. They did all the work.
Yeah. So all that remains is to say thank you very much, and on these occasions it's nice to go a bit beyond the normal Zoom protocols, and let's try and challenge the bandwidth a bit. And I'm going to ask everybody to unmute for a second and applaud. So thank you very much. And I think that probably that's the end of the meeting. So I wish everybody a good night. Thank you again, Tiffany.
Thank you.
Bye. Bye.
Bye.
Great.